You might be asking, what are we doing in front of this brick building with few plastic bags in our hands?
You ask yourself what the difference is? This one is based on fossil fuels and this one is made from biological matter. We use both of them in our daily lives, but they actually have a very different impact on our environment.
Right here at the TU Berlin, there is a different, very promising, sustainable bioplastic being developed called polyhydroxyalkanoate, namely PHA. And we have a chance to look inside the laboratory to see how it is produced. So come on, let's check it out.
Hello everybody, my name is Lara Santolin. I'm a PhD student currently working at the Bioprocess Engineering Department at the TU Berlin. I work in the group of Dr. Sebastian Riedl and we focus on PHA production.
Super cool. Could you describe what the project is about a little bit more?
Of course, just as a little motivation, we know that over 400 million tons of plastics are yearly produced worldwide, and we know that over 10% of this amount ends up in the seas, causing tremendous harm to the environment. For example, we know that every time we wash a single piece of clothing, over 2,000 pieces of fibers, synthetic fibers, ends up entering our seas. In this context, we work in a BMBF-funded project called PHAtex, which aims to develop a complete green bioprocess chain to produce novel, flexible and biodegradable PHA textiles. We at the TU Berlin deal with the PHA production itself. We work on strain engineering, on bioprocess development and on downstream processing. Then we send our polymer off to an institute in Aachen, ITA, and they produce our polymer fibers with spinning, and then these fibers are sent off to our industrial partners that do the knitting tests for textile applications.
And could you describe how the process in this laboratory actually looks like a little bit?
Of course. So the bacteria we use naturally accumulate this polymer as intracellular carbon and energy storage compounds, and it accumulates the polymer when it's faced to unbalanced growth conditions. This means that, for example, when they are faced to oxygen limitation or some stress due to nutrient limitation, whereas the carbon source is present in excess concentration, the bacteria accumulates the polymer. What we do is we get our bacteria, we grow them in stir tanks reactor. For example, the one I have in my background is a 100-liter bioreactor. We grow our bacteria under nutrient-rich conditions until we attain a certain cell density. Once this high biomass is achieved, we trigger nutrient limitation. In our case, we trigger nitrogen limitation, and then the bacteria start to accumulate the polymer. Once the bacteria is filled up with the polymer, we harvest our cells from the bioreactor, we centrifuge them down, dry them through lyophilization, and then we have to extract the polymer. What we do is we apply some sort of cell disruption, and then we dissolve the polymer in a non-polar solvent, and then we precipitate it out in a polar solvent, for example methanol. Then we have our solid polymer, we let it dry, and then it looks like this.
Oh wow, look at that.
So, could you tell me what kind of bacteria is used in the process? Are they recombinated or modified to make the process more efficient?
Of course. So, polyhydroxyalkanoates were first discovered around 1925 in Bacillus megatherium, but nowadays there are over 300 species of bacteria known to accumulate this polymer. In our laboratory, we use Ralstonia tropa, which is a gram-negative lethal autotrophic bacteria, and this is the model organism for PHA production for several reasons. The first one is that it can accumulate the polymer up to 90% of its cell dry weight. It can grow up to very high cell densities. It uses a wide range of feedstocks, and it also can be genetically modified to produce different types of PHAs. So PHAs are divided into short chain length, medium chain length and long chain length, polymers depending on the length of the side chain. And the wild-type strain of Ralstonia stropha can only produce the short-chain length polymer polyhydroxybutyrate, short PHB, but this polymer is very brittle and crystalline. The recombinant bacteria we use at our laboratory was engineered at the MIT and was brought here by my supervisor, Sebastian Riedel.
Wow.
And this genetically modified organism is able to accumulate the copolymer polyhydroxybutyrate co-hydroxyxynoate, so PHAtex, under feeding of fatty acid containing feedstocks. And this polymer has properties similar to low density polyethylene, which makes the application of the polymer huge.
So, and what's the most difficult part of the process?
So in PHA production we have two major bottlenecks. One is the cost of the carbon feedstocks, which represents up to 50% of the final cost of our polymer. And the second one is the downstream processing, which is very cost intensive and thus needs a lot of solvents. So we are tackling down both of these bottlenecks in our project PHAtex. Regarding the carbon feedstock, we are approaching strain engineering techniques in order to make our bacteria more substrate flexible. In particular, we want to engineer pathways for the utilization of sucrose and xylose. Both are available under low prices and big amounts in the market. And also we have another project in the group which is called PHABio-up, and there my colleagues are working in establishing a PHA production using waste animal fats, which look like this. Waste animal fats are waste streams of the slaughtering production and thus are also very cheap. Regarding the downstream processing, in PHAtex we are also working to develop a green purification process based on surface absorption, which doesn't need organic solvents.
It is known that petroleum-based plastic is very cheap at €1 per kilogram. Are you able to compete with that price?
So we produce really worldwide, as I said before, over 400 million tons of plastic. Only bioplastic account for only 1% of this total amount, and PHA commercial production only represents 50,000 tons a year. So, on the contrary, petroleum-based plastics are produced on a very large scale and thus have this low price of around $1 per kilogram. Nevertheless, this plastic causes tremendous harm to the environment, and there are also studies reporting that they biomagnify up the food chain, so we end up eating the microplastic that is in our groundwater and in our food. On the contrary, PHAs are fully biodegradable to CO2 and water, and they are produced with renewable resources. And petroleum-based plastics obviously not. So the commercially price of PHA is nowadays 5 times higher than petroleum-based plastics. We can work to reduce this price and to sink it down to around $2 per kilogram, but it is very difficult to compete with this very, very low price of petroleum-based plastic. So we do need political decision makers to show a clear position about this, to ban the boundless usage of petroleum-based plastic and to encourage the development of the bioplastic industry. Moreover, we also do need to revise our plastic consumption patterns, because there's no way the bioplastic industry will cope with 400 million tons of plastic a year. So we do need to reduce and do need to recycle more plastic, on site of producing more PHAs.
Thank you, that sounds very promising.
Thank you.